In this lecture, we are going to discuss the integumentary system. The word integumentary or the word integument meaning to cover, something covering. The skin. The skin is the largest organ in the body as it represents about 16% of the total body weight. It's formed of two layers. The superficial layer is the epidermis, which is stratified squamous keratinized epithelium, and the deeper layer is the dermis. Beneath the dermis, or the dermis rests on a subcutaneous tissue, subcutaneous adipose connective tissue, which is called hypodermis. This here is the hypodermis. Hypodermis is not considered as a part of the skin. It is not a part of the skin. It is helped to attach the skin or the layers of the skin to the underlying tissue. So we have here the epidermis, stratified squamous epithelium, keratinized, and here we have the dermis, which is connective tissue and divided into two parts. And here is the hypodermis, which is adipose connective tissue, and it is not considered as a part of the skin. First, we are going to discuss the epidermis. As I said, the epidermis is formed of stratified squamous epithelium, keratinized. This is the epidermis, formed of stratified squamous epithelium, keratinized. It is formed of two types of cell, or two categories of cell. First is the keratinocyte, and second is the non-keratinocyte. Non-keratinocyte is formed of melanocyte. This is the melanocyte. And Langerhans cell, this is the Langerhans cell, and the Merkel cell, this is the Merkel cell. As you see here, the keratinocyte arranged in multiple layers. This is the layer of the epidermis. The first layer, which is resting on the basement membrane, and it's called the stratum basale. The second layer is polygonal layer called stratum spinosum. Then the cells start to be flattened here and contain granules. This is stratum granulosum. Then a translucent layer called stratum lucidium, and the last layer, or the fifth layer, is stratum corneum. As I said, keratinocytes are arranged in 5 layers of the epidermis. Now I'm talking about the epidermis layer of the skin, and as I said, it's formed of keratinocyte, which is arranged in 5 layers. The basal layer, which is resting on the basement membrane, single layer of columnar cell called stratum basale, or the basal layer. Then multiple polygonal cells called stratum spinosum. After that, the cells start to be flattened and contain granules, so called stratum granulosum, or the granular cell layer. Then a few layers of cells, translucent refractile acidophilic layer called stratum lucidium, and lastly, or the fifth layer, is the stratum corneum, or the horny cell layer. The first layer of the epidermis is a stratum basale, or basal cell layer. This layer is a single layer and resting on the basement membrane. The cell here appear columnar in shape, single row of columnar cell with basal oval nucleus, and the cytoplasm appear basophilic. Why it is basophilic? As it is contained numerous ribosomes, as it contains numerous free ribosomes. Why? Because this layer represents or responsible for the regeneration of the above layer, or the superficial layer. So it is rich in free ribosomes, as it is considered as a stem cell for the epidermis. This layer, the cells of this layer, considered as a stem cell for the epidermis. In addition to the ribosomes, it contains also keratin filaments and melanin granules. Layer number 2, or the second layer, is called the stratum spinosum, or the other name is Prickle cell layer. As you see here, the cell appear polygonal in shape, This layer formed of multiple cells, multiple polygonal cells with central vesicular nucleus. The cell reach in keratin filament, and also it started to form granules called lamellar granules. What is the lamellar granules? Lamellar granules are lamellated granules, membrane-bound granules, I mean surrounded by a membrane, contain lipid and lipoprotein. This lipid or this lipoprotein liberated outside the cells in the intercellular spaces here to form a barrier. This barrier is waterproof for the skin. I mean it prevents the dehydration or the escape of water from the skin. As you see here, the neighboring cells are attached to each other by desmosomes junctions. Layer number 3, or the third layer, is a stratum granulosum or granular cell layer. This layer is formed of 3 to 5 layers of flattened cells. In this layer, the cell starts to be flattened and contain two types of granules. Either keratohyaline granules, is not surrounded by membrane, non-membrane bound basophilic granules. These keratohyaline granules contain a material or protein that bind the keratin filament together to form bundles, keratin bundles. The other type of granules present in these cells is the lamellar granules, which I mentioned before in the previous layer or in the stratum spinosum layer. The layer number 4, or the fourth layer, is a stratum lucidum, or the other name is clear cell layer. It is important to know that it is present only in the thick skin. Again, stratum lucidum is only present in the thick skin, so it is a point of difference between thick skin and thin skin. These appear as a translucent acidophilic layer formed of flattened cell. Here the cell starts to lose its nuclei and organelles, and only backed by keratin filaments. This keratin filament embedded in electron-dense matrix. The last layer, or the fifth layer of the epidermis, is a stratum corneum layer or horny cell layer. This layer, as you see, the very thick layer, is the most superficial layer, formed about of 10 to 20 layers of squamous keratinized cell. The cells here completely lost its nucleus and organelles and become fully keratinized or fully packed with keratin filaments. Again, I said the epidermis is formed of keratinocyte and non-keratinocyte. The keratinocyte arranged in in 5 layers, which I just mentioned, and non-keratinocyte are 3 cells: melanocyte, Langerhans cell, and Merkel cell. The first cell is melanocyte. Melanocyte present in the stratum basale layer, resting on the basement membrane. By light microscope, it appears with rounded cell body and arise from the cell body cytoplasmic processes. This is the cytoplasmic processes which branch in between the keratinocyte cell, branch or present in between the keratinocytes. It has central vesicular rounded nucleus. Its cytoplasm contains tyrosinase enzyme. This enzyme is responsible for the formation of the melanin pigment, so it is positive Dopa reaction. Dopa reaction is a reaction that proves the presence of tyrosinase enzyme. By electron microscope, melanocyte reach in mitochondria, rough endoplasmic reticulum, and Golgi apparatus, and this organelle is important for the tyrosinase synthesis. In addition to these organelles, we have melanosome. What are the melanosomes? The melanosomes are bound melanin granules. Granules surrounded by membrane contain melanin pigment in different stages of development. As we see here, this is the melanocyte resting on here the basement membrane, and here the stratum basale cells or keratinocyte of the stratum basale. And we have here the branches, cytoplasmic branches of the melanocyte present in between or branch in between the keratinocyte. we have here by electron microscope mitochondria, rough endoplasmic, and Golgi, in addition to membrane-bound melanin granules. I mean the granules which contain melanin pigment, and it is also called melanosomes. What are the functions of melanocytes? Melanocyte is responsible for the melanin synthesis. First, the melanocyte synthesize tyrosinase by help of mitochondria and Golgi and the rough endoplasmic reticulum. Then this tyrosinase enzyme is converted the tyrosine into DOPA. Then this DOPA polymerized into melanin and stored inside vesicles called melanosomes. So we have tyrosinase enzymes, which is synthesized by rough endoplasmic reticulum and then modified by Golgi apparatus. This enzyme converts tyrosine into DOPA, and then DOPA polymerize or collected together to form melanin pigment inside vesicles called melanosomes. After that, the neighboring keratinocyte, the nearby keratinocyte, started to phagocytose the tips of this of the cytoplasmic processes of melanocyte, which contain the melanin, and this is called cytokine secretion. So that the melanocyte deliver its melanin content to the neighboring keratinocyte by cytokine secretion to protect the neighboring keratinocyte from the ultraviolet ray radiation. This ultraviolet ray can damage the DNA, producing mutagenic effects. I mean several types of cancers. We have here the blue one is the melanocyte, and here we have the cells keratinocyte of the stratum basale, and here the stratum basale, and here the stratum spinosum. First, a tyrosinase enzyme is synthesized here in the rough endoplasmic reticulum, then transported to the Golgi apparatus to be modified, and tyrosinase liberated here in melanosome. Melanosome represent different stages of melanin synthesis. First, the tyrosinase acts on the tyrosine enzyme to be converted into DOPA. Then DOPA polymerized to form melanin pigment. After that, this is melanin granules. After that, the keratinocyte here phagocytoses the cytoplasmic processes with its content of melanin granules, and here is the melanin granules arranged above the nucleus. Here arranged above the nucleus, melanin pigment arranged above the nucleus in the keratinocyte to protect the keratinocyte or the nucleus of keratinocyte against the damaging effect of ultraviolet rays, which can cause mutagenic effect. Again, as I said, we have the epidermis formed of keratinocyte or non-keratinocyte. Non-keratinocyte, either melanocyte or Langerhans cell or Merkel cell. We have here the Langerhans cell. This cell present in the stratum spinosum layer here. This is the Langerhans cell. What is the function of Langerhans cell? It acts as antigen presenting cell. The third cell of non-keratinocyte is the Merkel cell, which is located in the stratum basal layer. As you see here, this is the Merkel cell. Merkel cell acts as mechanoreceptor. As you see here, this is the myelinated nerve fiber. The myelinated nerve here lose its myelin sheath, which is the blue color, and become in contact with the Merkel cell to form Merkel receptors, which is responsible for light touch sensation. Again, we have here the layers of the skin. The superficial layer is the epidermis, and the deep layer is the dermis. And we have here the hypodermis, which is not a part of the skin, which is a subcutaneous fatty tissue. Regarding the epidermis, as I said, it is stratified squamous keratinized epithelium formed of two types of cell or two categories of cell, keratinocyte and non-keratinocyte. Keratinocyte arranged in this 5 layer, stratum basale, Stratum Spinosum, Stratum Granulosum, Stratum Lucidium, which is only present in the skin, and lastly Stratum corneum. And we have underlying the epidermis is the dermis. This is the dermis, and you see two grades of color, this one and this one. The superficial part of the dermis is called the papillary dermis, and the deeper part is the reticular dermis. The papillary dermis is more cellular than to be fibrous, contain more cells than fibers, and the reticular layer, the reverse, contain more fibers, collagen fiber type 1 and elastic fibers, and less cellular. Papillary layer, more vascular to supply the avascular epidermis. The papillary layer contain more blood vessels and more capillaries to supply the avascular epidermis. In the papillary layer, we have receptors, myasino receptor here. This is myasino receptor, and the reticular layer contain Pacinian receptor. To compare between papillary and reticular dermis, it is a very important issue and a very common question. We have here the dermis. This is the comparison between the papillary layer and the reticular layer. As I said, the papillary is the superficial layer which is forming the dermal papillae. The reticular is the deep layer. Papillary is more cellular and less fibrous. On the reverse here, the reticular is less cellular and more fibrous, contain collagen type 1 and elastic fibers. The papillary layer is more or highly vascular, as it supplies the vascular epidermis. Regarding the receptors, we have here myasino corpuscle in the papillary layer, which is responsible for the touch sensation, and in the reticular layer we have Pacinian corpuscle, which is responsible for the pressure or act as pressure receptors. Lastly, we have two types of skin, and you have to differentiate well between both of them, and this table is very important also, and is considered as a common question. The site of thick skin present in the palm of the hand and sole of the foot. Thin skin present all over the body except these two sides. Regarding the epidermis, in the thick skin, it is formed of the 5 usual layers which we have mentioned. Again, it is a stratum basale, a stratum spinosum, stratum granulosum, stratum lucidum, and very thick stratum corneum. In the thin skin, we have only 4 layers. No stratum lucidum is present here, and the stratum corneum is a very thin layer. Regarding the dermis, we have here in the thick skin more sweat gland, more numerous or more abundant sweat gland. In the thin skin, it is less abundant or less numerous, few in number. Regarding the hair follicle and the sebaceous gland and the rector pili muscle, here in the thick skin, the 3 are absent. In the thin skin, the 3 are present, as this considered one unit, the hair follicle, and the sebaceous gland attached to the hair follicle, and the rector pili muscle also attached to the hair follicle, so it is considered as one unit. So these 3 items absent here in the thick skin and present here in the thin skin. This is a photograph, or it's a section for thin and thick skin. We have here the thick skin, and we have here the thin skin. You can notice the very thick stratum corneum here and very thin stratum corneum. We have here numerous sweat glands. This is sweat glands. This is the sweat glands. And we have here the hair follicle. This is the hair follicle. And this is another one. And this is the sebaceous gland. This is a sebaceous gland which is absent here. Thank you for your attention.